Okay, hello everyone, and thank you for kind invitation from Irina and Rob. So I guess I'm speaking to a very diverse community, so don't hesitate to interrupt me if I'm kind of losing your focus, because there will be some technical details as well as some applications in biological systems. So I made it a little bit like broader overview and a little bit of the historic perspective. So I will start with introduction of methods and techniques of X-ray emission spectroscopy. And what is good about this technique? It is high energy resolution, and we are talking about recording spectra with 0.1 to 1 eV resolution. And if I don't forget, I will try to comment as we go along on that technical characteristics of resolution of the spectrometers. But, nothing comes for free. So you want a high resolution spectra, you have to kind of throw away a lot of photons. So it's a photon starved technique. So high intensity X-rays are important here. So this technique never been historically done at bending magnet. It's been always requiring some form of ID beamline. And we'll talk a little bit about the spectrometer. So we will compare Johansson and Van Hammer's techniques. And this goes back to like beginning like 1930s of the previous century. So X-ray optics has been well understood already at that time, and people could make analyzers. Biological applications came significantly later with kind of hard X-rays and third generation synchrotrons, and I'll show you. So when we talk about the biological, we talk about dilute and radiation damage sensitive. Dilute is sub-millimolar concentration of the metal of interest, so can be also significant backgrounds. We will also talk about it. And when you have something photon starved and very dilute, and then you try to make a time result, you lose another factor in your signal to noise, because you can only collect data when the X-ray pulse is present. And sometimes the function of your biological system can limit the repetition rate, etc. So, and I'll try to present result. Some are published, some are in progress, and we just finished another beam time a few weeks ago, so I'll show some most recent results and kind of discuss the outlook. Okay, so for very broad audience, what is X-ray fluorescence? X-ray fluorescence is like optical fluorescence. So you get photon in, you excite your system into a certain state, and then eventually the system will relax and give you a photon out. Do you see my cursor on the screen? Or should I make it like a pointer?
I personally see the cursor.
Okay, so then it's enough, yeah. Okay, so photon comes in, excites, photons comes out. So everyone knows it from optical spectroscopy. So here we're talking about X-ray photon. So X-ray photon can be very hard, the high energetic X-ray photon. It can completely ionize your atom. And we are talking about, like you see here, is X-ray fluorescence for different elements, and some are biologically significant, such copper, zinc, manganese, iron we will be talking about. Yeah, so you can ionize electron from 1s orbital into continuum. And then you have various orbitals, 2p, 3p, etc. Various orbitals from where electron can repopulate this state with emitting of the X-ray photon. And there are certain probabilities of all those different processes. And of course, we know all quantum mechanics, they will occur at different energies, etc. So when you excite your sample with the X-ray, you have this ability to record X-ray fluorescence. Is very broadly used for so many different applications. And here you see the typical spectrometer, and its energy resolution is about 100 eV. So you remember the previous slide, we started talking 0.1 to 1 eV versus 100 eV. So with this device, you can record that fluorescent photon, but the broadening of the spectrum is significant. But still, you can do certain very useful analyses already there. So now, what is our motivation? So we want to record with high energy resolution because, so this is a kind of more expanded, more involved diagram of all those energy levels in your atom of interest. This metal atom can sit inside the protein, or they can be a cluster of metals inside the protein. So we want to know metal oxidation state, for example. You know, metal in the protein is there for a reason. So usually it's a lot of iron-containing proteins that do all the fancy, unbelievable oxidation reactions in the organism, make a chemical synthesis of very unusual molecules, etc. There is this manganese I will introduce, which produces all the oxygen in the atmosphere. So the metals inside the proteins, they can do amazing things. And metals, as we know, we can be iron 2+, iron 3+, iron 4+. The same is for manganese. And there is also some copper-containing enzymes, which also do like methane monoxygenase. They can make from methane methanol, so many interesting things. Iron nitrogenase, which can fix nitrogen to ammonia. So all those interesting things. So we want to understand basically the electronic structure, everything including oxidation state, how many electrons, how they move, how they distributed on those orbitals, what is the ligand environment, because you understand the ligand environment will change levels of those orbitals, etc. So basically, doing this kind of type of high energy resolution X-ray emission spectroscopy, you can completely map out every energy level in your reaction center. And when you map out every energy level, you kind of understand the system. So let's talk a little bit about terminology. So this is the absorption. Absorption can be resonant when it goes into a certain level, or can be non-resonant when you're just simply ionizing the system. And then emission can be from the different level, emission from 2p to 1s called K alpha line, from 3p to 1s K beta line, and then valence to core. Also, I'll show you examples where this tells about the ligand to the metal, and they could be, you know, other types of things. So as you see, the scheme is very involved, and there are many transitions. If you have high energy resolution, you can record and learn about them. So for example, the first kind of most probable transition is from 2p to 1s, so those are the most intense line. And as you see here, we just draw one level, but it's not one level, it's split by spin-orbit coupling. So again, whatever you learn in quantum mechanics class is very, for some might be very long ago, 30 years ago, in the quantum mechanics class may be very relevant because it immediately represents here in the two peaks with a certain relative intensities, because 2p level is split by spin-orbit coupling, and so on and so forth. So there is a K beta line, which comes from 3p, and the valence level, which can tell about, so the valence level you can think of like electron from the ligand, like electron from the oxygen or electron from the nitrogen, jumps down to the metal 1s orbital. And this energy difference is very, very specific to whether it's oxygen, nitrogen, carbon, etc. So you can learn a lot of interesting things, and you can again like do high-level calculations and try to predict where those energy transitions should be and compare with your experiment. So I hope I motivated you enough to go into the pain of experimental realization of this technique. And this is the first example. So historically, people started with Johansson type of spectrometers, and I would call it monstrosity because they're pretty big. So, and when I was a postdoc, I started working with those pretty big, and they have 1 meter. So I have to go away from camera. 1 meter, so sample to analyzer distance. So they will be on the Roland circle, but 1 meter focus in length. So it's a monstrosity for which you have to have, like, you see, specialized table, specialized place, and so on and so forth. So, and also it has a very expensive spherically bent crystal. So basically, this is Johansson type spectrometer, involves a spherically bent crystal analyzers, and they focus X-ray emitted from the sample. of specific wavelengths. So if you fix, as you see here, there is a sample, a detector can be above or below the sample. If you fix this angle, so you see this purple angle, you're reflecting only one energy within, like, about 1 EV bandwidth. So the energy resolution of this kind of spectrometer will be 1 EV approximately. And then to reflect another energy, like plus minus 1 EV, you have to change this angle. So it's not just a monstrosity, it's also monstrosity that you have to continuously move if you want to scan and record. So if you want to record this spectrum, and this spectrum takes 20 EV. So again, high energy resolution, maybe 1 EV is your instrumental resolution in this case. You have to move this entire huge assembly with the crystals and detectors on that, motorized stages, and God forbid if you start losing the steps or something on your motors, everything kind of, you have to start from scratch. So very, very difficult technique. I worked with it myself, so I know the pain. Let's see summary. Available at synchrotrons around the world. So I will show you examples, things are available, things potentially can be brought to a given beamline, but it's very, very painful. It's a very long setup time. So this is how people started collecting biological data, data from biological samples from proteins, because this has a good background rejection because of that very expensive spherically bent analyzer focusing everything into one point. You can collect biological data. It suppresses background greatly. So the benefit is suppression of the background, good background reject, not suitable for time resolved. Again, people use it for time resolved because you have to have this scanning. You can't get the full spectrum in one shot. Okay, so how this whole historical, so now I'll start to show some data. But before I start to show the data, I'll introduce briefly photosystem 2 because a lot of data will be for photosystem 2. So photosystem 2 is a transmembrane protein complex. It's involved in photosynthesis. There are two kind of green proteins there, photosystem 1 and photosystem 2. They contain a lot of chlorophyll molecules. That's why they are green. And they have some kind of electron transfer chain that eventually can split the water molecule. And in the center of it, there are 4 manganese atoms and 1 calcium cluster, or 4 manganese atom and 1 calcium ion. They are forming the cluster. So a lot of things are now known about it. But the motivation again, so through the catalytic cycle, learn about changes in this manganese ligand environment, oxidation state, and how the catalysis proceeds, and the bigger motivation that we can kind of steal this process and use it for clean energy production. So the catalytic cycle is very complex. So here you see, this is the dark stable state where everything starts. It has manganese 3 plus, manganese 4 plus ions. And you can luckily clock it with the laser pulses, and you can give 1 laser flash, 2 laser flashes, and then after the 3rd laser flash, some kind of transient process happening, and the clock time is 1 millisecond. So the slowest step between S3 to S0 takes 1 millisecond. So you cannot measure the system quicker. Like if you want to study this catalytic cycle, at least you have to have a certain rep rate, which is not great. Like you can't do kilohertz probably. So you have to observe this 1 millisecond catalytic step. So in certain sense, this is not very fast protein. So it does 1,000 times per second. But in certain time result techniques, 1 millisecond is still fast. So for example, if you say, oh, like I'll prepare my protein in S3 and then I try to freeze quench, it will be very difficult to freeze quench. So you really need to, so those reactive intermediates can be trapped. Those, they have to be observed in a system. Okay, so again, we have this very complicated protein, 500 kilodalton. a lot of cofactors, a lot of functionality, and we want to know structure, ligands to manganese, redox states, spin states, mechanism for oxygen-oxygen bond formation. Because somehow magically it can accumulate. This is a 4-electron reduction or 4-electron reaction. It's oxidation of water. So you have to accumulate 4 holes. So basically there is a photo-induced process which removes electrons from the cluster one by one. So after you removed 4, you magically combine 2 oxygens into O2 and then thing restarts. Okay, so, and there was a lot of interest. So as you see, biological applications kind of kicked off around 2005. We published the first paper. And this experiment was done actually at BioCat. BioCat, if you remember, Raul Barrera was still there. So basically we had 2 weeks of beam time. We came with this monstrosity table for the spectrometer, liquid helium cryostat. I think if I want to do this experiment now, I would be bankrupt just by amount of liquid helium I need for 2 weeks of beam time. We came with thousands of protein sample, very thinly filled because the penetration depth about 0.5 millimeter. So concentrated, thinly kind of filled samples in the sample holders. They were loaded into the liquid helium cryostat and they were shoot point by point with the focused X-ray beam, maybe like under 1 second exposure per point. So that's why so many samples. And then, you know, this is just a tiny cartoon here, but the spectrometer array was a monstrosity and took several days just to set up and align all those spherically bent crystals. So very difficult experiments. And in this case, we did not even move the spectrometer. We did not even record that high energy resolution spectrum. We were simply sitting at the top of this manganese K alpha line. And instead we were sweeping this monochromator and we were collecting so-called high resolution EXAFs. So that allowed us to clean up data from the iron, which is always part of the protein, and we could record kind of high distance resolution for manganese-oxygen, manganese-manganese interactions. And based on this information, we could model. So we can come up with the model of the cluster and position it in a certain orientation in the membrane and kind of eliminate certain models. So it was prior to when people knew the arrangement of atoms in the cluster, prior to high resolution X-ray crystallographic data on this system. Okay, so here again, we are only sitting, we like aligned everything and we are not moving, we are collecting, we're instead changing monochromator. And this is a different version built by Uwe Bergman. So this is the attempt to miniaturize that huge table. So instead of huge table, now you have a huge disk like a dish, and you have a lot of these motor controls because all of these crystals, they're kind of movable by those motor controls. So it's a kind of miniaturized, but not quite. And then with that setup, we were able to, so this is the bottom one, electronic structure changes. So we were able to scan the spectrometer and also scan the monochromator and record the so-called RIX, resonant inelastic X-ray scattering data. And this is S0, S2, S1, S2, S3 data, and the top is manganese reference compound. So, and we were trying to figure out all those electronic transitions, but I think here, as you see, blobs don't really differ that much from each other. So it was another like 2 weeks of beam time with thousands of samples, but you know, it was only paper in inorganic chemistry after many years of data analysis, because from your eye, you see blobs don't really change much. And maybe it's again like 1 EV resolution, maybe the higher resolution is needed, maybe some other lines should be observed. And then we also had another paper where we detected this metal to ligand transition. So I can go back to this very basic diagram. So basically we detected this thing in photosystem 2, and we showed manganese oxygen interactions. We could estimate the distance, everything was in agreement, but again, it was only done for S1 state, for one state. Okay, so basically you can do it with biological samples, with the cryostat, with very bright X-ray beams. And here there is another example of that. So basically I stop here with direct detection of oxygen ligation in this cluster, and exactly the same experiment was done for another protein. So it's again SSRL and this kind of dish, Johansson type spectrometer, similar to that dish. And here the Serena De Beers and her group, they analyzed nitrogenase. And nitrogenase is also a very fascinating system because it can take nitrogen, which is very inert molecule, and make it into ammonia. And crystallography was unable to resolve the central atom. So, and by doing that very, very weak signal, this valence to core transition, they were able to say this is carbon. And I think since then that has not been objected. I'm interested about assembly of that thing, etc. So it's still a lot of interesting questions here, but I think that's the only way how you can detect this carbon. If you know of a different way, let me know in the discussion. Okay, European efforts. So there is, I talking about efforts in APS and SSRL. ESRF also has its own effort. They have a Peter Glatzel, and he has also another monstrosity thing, but it works very well. So, and as you see, here are the publications from that equipment. So they have manganese K beta and manganese K alpha, about 1 eV resolution, 1 meter Roland circle. And they attempted kind of early attempts of time-resolved analysis of photosystem 2. And using the fast shutter, so fast mechanical shutter has a response time of 1 millisecond. So now you immediately compare 1 millisecond to the transient process of 1 millisecond in that catalytic cycle. So basically you can see that he can detect all other states like S1, S2, S3, which are kind of longer lived, but he does not have a time resolution into that transient process. And there is other publication, K beta, K alpha lines. And here is the high energy resolution again for like this last paper is, you see 2025. You see it again on the top of this K alpha line, and you scan you do Xens, and you just kind of sharpen your lines a little bit for a more detailed analysis. Okay. So those are all the kind of pre-time resolved time and all Johansson spectrometers. So now we transitioned into the era of free electron lasers. And for free electron lasers, people realize that Johansson is not good because you don't want to move things. You want to do a dispersive detection. And people change to so-called von Hammer's geometry, and you have a cylindrically bent crystals. So you focus on that cylindrically bent direction, and you disperse on the flat direction. So again, the physics behind those spectrometers is very simple. It's just a Bragg diffraction. So any undergrad knows Bragg law. And so basically in this case, you diffract, like you have a source and you have a crystal, and you diffract, it's like a prism in some sense, but for X-ray. So you diffract different wavelengths into different position. But then, if with Johansson, you can have a simple photodiode type detector, like one element silicon detector. Here you need to have a position sensitive detector. So basically, you don't need to scan. You can be in a fixed geometry, but you have to have a position sensitive detector. So the more sophisticated detector is needed. Okay, so how this thing was realized at LCLS. LCLS has another monstrosity, so I worked with it also. It's pretty big, and it has a good number of crystals, and each crystal is also very expensive. And I don't know, 12, 20, some good number. So this is a photo from the back. And this is the example of manganese K-beta line dispersed on the position-sensitive detector. And, we did some work there too. We published and etc. So in parallel, okay, and from this detector, the representative X-ray emission spectroscopy for PS2, very interesting S2 to S3 transition where this final oxidation happens. This mostly oxidized S3 state is formed before the transient step. The results are published. First moment analysis of this K-beta line tells you how manganese gets oxidized. You see number of time points were detected, and here the structural change in the cluster is combined with the change in oxidation state on a certain, like you can try to match, when this extra oxygen gets bound versus when manganese gets oxidized. So you can kind of combine structural and electronic changes information in one experiment, but you see the year, it's already 2020. So it's kind of came significantly later after those initial studies. So this is big von Hamas, cost about 300,000 at LCLS. I think people can write beam time proposal and use it. Again, every crystal has to be tuned, and multiple crystals get lined up into the same line. So when you see this line, it's several crystals focused onto the same line. So you can never do this perfectly. So you are losing some energy resolution in this process, or like you gain some heterogeneity. So it's about half EV energy resolution for this technique. Okay, so there is a representative data. And in parallel, I had this very enthusiastic, very talented student who is now a professor, I think up for tenure this summer, very soon. Okay, and she developed some completely in collaboration with Jerry Seidler, completely different idea of miniaturized spectrometer. So if you see this white box, you can kind of grasp the size of it. It's really very small. And this is the substitution for all those monstrosities you have seen on the previous slides. So it was really a kind of very eye-opening experience for us. And we started collecting time-resolved data. And here you see that system had some kind of 3D printed frame, and 3D printed frame contained simple flat crystals, but it was 3D printed with a certain geometry that they have a dispersive direction here. The dispersive, it means energy of the X-ray changes in this direction, and in this direction is a kind of focusing those X-rays, and we can collect a good amount of data. Again, we're gaining the background here because we're not that tightly rejecting scattering, and also the system is very dilute, but this is how this spectrometer performs. And we did some studies with this spectrometer, but then there was a push to improve it, to make it even tighter in terms of better background rejection and bigger solid angle to catch as much fluorescence as we can. And this came to these kind of boxes. So in this box is 0.5 meter von Hamas, so it's getting a bit bigger. But that's as big as it gets. So it's half a meter in one dimension box. And now we have several boxes for several very interesting biological lines that show you a table, and there is a procedure. So we pretty much manufacture those crystals ourselves. So the part of like why those previous Johansson and von Hamas are so expensive, because those curved crystals are very difficult to manufacture. They can cost easy like $10,000 one crystal. So here we, again, like with the help of Argon staff, we manufacture crystals ourselves. All our crystals are self-made. And again, like if you're interested, I can explain more details, but the procedure is on this on this slide. And again, like, because we're doing time-resolved spectroscopy eventually with the systems in our sample, we pretty much have to discard it after every X-ray shot. We have to have some sample delivery systems, and this is still also under development. The first generation, we used some kind of painted strings that we can translate very fast with the step motors. Here we have a drop-on-demand synchronizable with the beamline. Okay, so I think I spent another 10 minutes trying to walk you through the things we learned about the photosystem 2 time-resolved analysis in those years. And then I'll make sure, like last 5 minutes, I show you some new results. So what are the frontier in photosystem 2 research? So basically this research started in 1970s with the experiments of Koch and Juliot. They showed that the oxygen evolves with the pattern of 4 flashes, and that makes sense because it's a 4-electron process and we have to accumulate holes on that cluster, and magically oxygen bond will be made and oxygen will be released. Time-resolved X-ray diffraction is, of course, at the far front of those efforts. So there are crystals of PS2 diffracting to 2 angstrom, and people can do exactly the same timing schemes we are doing for the spectroscopy and try to see how the structure changes. I'll show you the recent results in comparison with spectroscopic results. Time-resolved X-ray spectroscopy, this is where my group is active, among other things, and DFT modeling. So we also try to do DFT modeling from time to time. So there was early data sets which were recorded, like basically full data sets were 2015, publication 2018 with one more additional data set recorded with this like really miniature small box, which cost basically nothing, 3D printing and cutting wafer into square pieces. So nanosecond laser at 530 nanometers, 1, 2, 3 flashes, X-ray. At that point, we could do 10 to 11 photons in 40 microsecond pulse at biocars. So this is all biocars data, and we are analyzing, and we were able in that within that 1 millisecond window, we were able to slice much smaller kind of time windows. Most interesting would be 200 microseconds and also 50 microsecond. We added like the earliest we could observe. And we found something very unexpected, that already in that transient process, spectra changing very early. So in 50 microseconds and 200 microseconds, they are already changing spectra. And by that time, there were no computational models or theory which could explain why this is the case. And also to kind of connect those small changes in the spectra with the chemistry. So we think that the S3 state, which is the last stable state between the transient process, it contains all manganese 4. It might contain manganese 4 double bond oxygen. It might contain manganese OH. It definitely contains some bridging oxygens. And if we to change any of those into a bridging peroxide or terminal peroxide, we will have oxidation of the oxygen and reduction of manganese. So we will have this electron redistribution between manganese and oxygen fragments. And that's what we think has happened and caused this change in the spectrum. Okay, so, and this is going into a bit more details, but again, like just interrupt me if I'm losing your attention. So again, photosystem 2 is very deeply studied system. So people tried everything possible on it. So there were some optical pump probe data where people tried to determine the dynamic of this tyrosine, redox active tyrosine, which takes electron from the manganese cluster. And of course, we have techniques to quickly monitor oxygen release. And already early, like late 90s, people knew that kinetics of this tyrosine reduction in this S3 to S0 in this last most important step, and release of oxygen. Very similar. So basically, whenever this guy gets electron, this system spits out oxygen. And that was called the kinetic challenge. And they say if this is happening, then oxygen bond formation has to be within 50 microseconds, which was a bit surprising. Then there were some very detailed computational methods, and they would contradict. They would say, you know, first you have to remove electrons. So first you are removing electron here, blue arrow. Then you have one barrier. Then you have another barrier, and only then maybe oxygen will come out. So this computational model contradicted these early experimental observations. And then they were also time result X-ray absorption. But as you see, so you saw there's pretty full data, and you will see more of this data versus these kind of, I would say, few data points, maybe like 10, 20 data points of the lag phase. So basically in this lag phase, in this point, we can collect the entire response, entire representation of electronic structure of the system, so which is more informative. But here in X-ray absorption spectroscopy, that seems to be like a lag phase, at least in those experiments. We were trying to redo those experiments with better detection of signal, but that was only partially successful. So basically we had limited number of people and resources, so we had to focus on something. Maybe eventually this will be redone at the modern level. So this is 2005. But yes, so this is all available information, and it's a kind of calls for a very special mechanism, so in combination with our data. And to explain our data, we had to propose that S3 state is a very special state, which is highly predisposed to oxygen-oxygen bond formation. So it almost has some kind of equilibrium between having oxygen-oxygen bond and not having oxygen-oxygen bond. And then environment of the protein where tyrosine is now oxidized and ready to pull the electron out, that change in the environment, maybe a little bit of change in coordination of amino acids around the cluster, triggers this transition. By some calculations, it can be a small energy difference as 0.2 eV. It can trigger this conversion, and then the last electron is removed from the peroxide. And that makes a lot of sense in terms of environmental adaptation, like evolutionary adaptation, because you don't want peroxides in your system. Peroxides in your system are too damaging for any biological fragment. So basically the system can make peroxide only when there is this oxidized tyrosine around it, and then the job is done with this final electron transfer. But this electron transfer is also eased because it's not oxidation, another oxidation of the manganese, which is already like pretty much fully oxidized. It's oxidation of peroxide, which is much easier. So it's kind of photosystem too is amazing because it works at the constant potential, while all other catalysts, they require higher potentials. It's a topic of different discussion and conversation, but basically we had time resolved data, we had a computational model to go with it, and that's how that kind of 2018 and Kate Davis PhD concluded. And since then, finally, we published in 2025. So we repeated it. So with the new sample delivery system, new spectrometer, kind of more sophisticated, slightly bigger, we reinvestigated this process in D2O. So because there is a lot of proton-coupled electron transfer step, the system has a clear kinetic isotope effect. So when you are moving electrons, but you're also moving protons, changing system into the D2O makes a difference, and you can, you can learn something about the process by measuring the system in D2O. And as you see, data quality improved, and we also had many beam times for that. So several beam times were merged for this data quality. And again, at the early times, we could see the change, and we could see the change at the later time. So there was a little bit difficulty with this publication because we did not see the change at 200 microsecond that clearly, but we were able to explain it with a certain kinetic consideration. So it still shows that these experiments are difficult to do. So what have we learned in the kinetic experiments or in D2O experiments is that we think this transient process, this most interesting kind of oxygen-oxygen bond formation step, is extended in time. So we saw the early onset of the reduction as an H2O. So this early onset of the reduction was not affected. So, but we think that the lifetime of this intermediate is extended based on our data. And that's why maybe if you want to structurally characterize the step, it's better to do experiments in D2O. Maybe people try to do crystallography in D2O, etc. But overall, the results were very similar. So now, as I promised, to compare and contrast the latest. Yeah, so I think to the best of my knowledge, the latest is this Nature paper 2023, which discusses the structural changes in the same time frames where we have spectroscopic data. And basically, this paper says, oh, nothing happens till like 500 microseconds. So there is maybe some proton motion, but nothing happens. But if I look at the data, so this data is from this paper. So basically, this is manganese 1, manganese 4, so 1 to 4 distance, and then 1 to 3 distance. So it's basically distances between heavy atoms, which hopefully you can measure precisely. So nothing happens in your system, but at 50 microsecond, you have, and here it's outside of the given error bars, you have a significant structural change. And after the structural change, this distance remains, again, like if I kind of average those 3 points, it remains elevated above that level. Here, the same thing. So again, like for everything, we have error bars in this method. But I would argue that at least the way I look at this data, already at 50 microseconds, something happens to the heavy manganese atoms, and this might indicate the chemical reactivity. The only now contradiction that comes, so we propose this mechanism of oxygen-oxygen bond formation between those two oxygens, but in this case, the DFT model shows that the distance between heavy manganese should decrease, not increase. So that's a kind of comes at the point. However, there is another model where you can also have a peroxide formed here from, let's say, water from the calcium. So it's a different, you either do radical coupling or you do OH, kind of a combination with this manganese 4. This model gives increase. So again, we do think the chemistry is happening at this early, like, 200 microsecond time scale. But if we are tracking the heavy atoms, maybe it's a little bit different type of oxygen-oxygen bond formation. Currently, we don't know, but as you see here, how spectroscopic results and structural results can be combined to build the model for the reaction. Okay, from this, I will move on. So as I advertised, we have now several boxes. So we build iron K beta and manganese K alpha boxes. If you want details, I can give it to you. I'm happy to collaborate, to come with my box or to give my box or come and help you with my box for your beam time. And if you use those for concentrated samples, that's a magic. So it's like so easy to use. And we published some papers for like battery materials, catalysts, fuel cell catalysts. So the biological application, I mean, iron K beta spectrometer was very much motivated by biological applications, even starting from some controversies in myoglobin, hemoglobin, tons of iron-based oxidation enzymes. So like I think that the space for iron-based chemistry analysis is like enormous. So we only did this one demonstration, which was also done at APS, not at BioCars, but just at ID Beamline. I believe it was sector 20. Beamline in collaboration again with Kate Davis group. So she is expressing and studying this very interesting protein, which has iron a kind of in the heme motif, and heme-dependent, protein, which makes a chemistry with basically synthesizing chemicals from tryptophan. So the tryptophan is a substrate, and there is a spin change. So with iron is a little bit tricky because iron 2 plus iron 3 plus doesn't have that much shift in this particular line. But if you change the spin, it has a big shift. So if you have a protein in where you think the iron is changing the spin state, that would be really a fantastic thing to study. Okay, and here is to the manganese K alpha box. So that's our latest box. And basically you see, till now, we had to have several beam times and combine the data to say something about the spectra. So now we want, theoretically, ideally, to have one beam time and record the data. And then maybe do a second beam time for reproducibility and then start doing more. More means more time points and time resolved dynamics. Cyanobacterial prep. So far we did only spinach, which is more accessible. Cyanobacterial preps give you little protein. You need to economize on your sample delivery. Maybe you want to study some mutants. There are some very interesting mutants. There is a strontium substitution. You can substitute calcium for strontium. So we want to study all those specific things, but how can we physically do it if we need like 4 or 5 beam times to have one complete publishable data set? So we looked at manganese K alpha. So our idea is, okay, manganese K alpha is a bit, it probably has smaller shifts, but it's more intense. So let's see how it will go. So this is our BioCarson published data, so latest beam time. So this is how reference, so this is concentrated sample, looks on the position sensitive detector. This is how PS2 looks on the position sensitive detector. You might see a little bit of those peaks. This is how you process the data look. As you see, single beam time, you already have a very decent data quality. And for the final, you combine those 3 crystals into one spectrum. So basically you can add up those 3 spectra, and the data pretty good. But as you see, again, it sits on this huge background. If we could reduce this background, we would be even much more happier. We're doing shielding and, tight apertures and everything is calculated. But again, you're shooting at 0.5 millimolar manganese in membrane, in protein, in water, so everything scatters. So in the idea, that we can do more beam times, go to more places. And again, while APS was undergoing upgrade, we got this beam time at ESRF, like we applied long ago. So finally, because they had to improve their beam line to be able to even invite us. So they finally did all those improvements. And this is the same experiment with the same box done at ESRF. Beamline 09, and this is different detectors. So this is Jungfrau 1M, and you see European people, they kind of connected with this Jungfrau project that's been around already for at least under 10 years in different, and this has several modules, so it's a pretty big. We don't need that big. We can have just 1 or 2 tiles enough. And this is the concentrated MNO reference spectrum. But what they can do, which Pilatus cannot do, they can produce this energy spectrum as well. So whenever photon, and you have to run detector in a kind of also photon-starved regime, but with our PS2 is not a big issue, so we're always photon-starved. So if you have, like on this 1 million pixel detector, not more than 1 photon per pixel when you kind of do your acquisition, you can do this processing. And you can sort out, you have some information about energy of that photon. So, and this is, as you see, excitation at 11 keV, and this is energy. This is energy in keV approximately. So this is the kind of, what they say, it takes a lot of computational power. So basically you run this detector with some kind of big server, which kind of sorts those photons, understands those X-ray photons for you. when it is done, you have this distribution. So this is concentrated sample. So you see manganese somewhere here around 5 to 6 keV. You have your scattering, we excited 11, maybe some germanium fluorescence. The spectrometer has germanium crystals. And then there are, high harmonics that we were discussing how to clean. And this is the spectra. So nice and clean, concentrated sample. Now we go into PS2. So if we would have no energy sorting, okay, and then based on those plots, you can put the energy cutoffs. So you say, I don't look at the photons if they are more than 6.5 keV. So I reject all the scattering, which is not manganese fluorescence. So basically, because this beamline has certain shortcomings in comparison to biocars, at least how we understood it, like high contamination of second harmonic, etc. So if you don't do any energy filtering on the detector, you have picture like this. So PS2 is impossible to see. So if we would come there with the Pilatus, we would just, pack everything and go back because we would have no data whatsoever, or, we would have this very poor signal to background data. So basically, this is no energy filtering, how detector looks. This is how spectra look if you don't do any energy filtering. However, this is after energy filtering. Then you say, I ignore all the photons which are above 6.5 keV. And it really works. So you see how much cleaner. So you start to see your manganese of the PS2, and you start to see, spectra on top of the background. So basically what I'm trying to say, if tomorrow I can get this detector to the biocars and get from this spectrum, I can probably cut half of that background down and basically collect my data better, have a better quality of data, need less protein for my data. So I was kind of excited. So basically we got some data there, not spectacular because of other reasons, but I see kind of a light at the end of the tunnel, how combination of spectrometer design and new detectors can make time-resolved X-ray emission from the very exclusive niche to a more mainstream technique, which can be straightforward combined with the crystallography, because if you see the box, the back is fully open, so you can still deliver a crystal and you can shoot diffraction in the back, nothing prevents you. So maybe you have a little bit of shadowing, we should try. Okay, so with this, I have my conclusions. So I hope I convinced you that this method can deliver critical information on metal oxidation state, spin state, ligand environment, and those are all central to understand the mechanism and functions of metal cofactors, because any crystallography experiment will still have a heavy element blob, but to see kappa 2 plus, kappa 1 plus, maybe you need to do some anomalous below the edge. So again, you have another, crystallography has very limited tools to answer those questions. Photon start techniques, that's why it is so difficult, but with high brilliance of new energy X-ray sources, it's becoming more and more available. Time result realization is straightforward. Actually, we never had problem with timing and delivering X-ray pulses. Triggerable sample delivery is another big problem. So if you have a more economic sample delivery system, I'm very happy to connect and discuss. And as you see here, we were talking about this, rejecting the background. You saw that Johansson technique has a good background rejection, but it has other shortcomings of very, very complex instrumentation and very new emergent. I think that's the first demonstration of energy filtering on the detector for X-ray emission application at the synchrotron. So with this, I would like to thank you for attention and acknowledgement. So it's my lab. And Irina and Rob been with us, I think, since 2012, the most valuable long-term collaborators. You see Steve Hild and Dale, they transitioned to happy retirements, but Irina and Rob, they're still going strong. And yeah, so our new kind of new people who were adventurous enough to try to schedule beam time with PS2, time resolved beam time with PS2 is Matteo and Marco. Okay, I'll be happy to take any questions. Thank you for your attention.
Thank you, Yulia. Very interesting talk. As Yulia said, are there any questions? I guess I have a quick question about, you said you used the spinach for your sample, right? I guess the deuterium, is it easy to do a deuterium exchange then, or with that?
Yes, it's a deuterium buffer. So we are not deuterating the protein. So
Okay,
the
that's what I
protein
figured.
steals all with hydrogen. So the spinach is normal supermarket spinach.
Okay,
But
there's
we're
nothing special.
exchanging, yeah, in the water because the proton coupled electron transfer. So it's a lot of things in questions in PS2. I'll just get this general cartoon I have here. So a lot of questions because you have those bridging oxygens, maybe you have terminal waters. No one knows the protonation state. So even after all this high resolution crystallography, they say this manganese 4 might start with 2 waters or 1 water, 1 hydroxide, 2H, like no one knows. So if we could even figure out those protonation states, it will be already a big step forward. But with, because there's like waters and OH, they exchange protons really quick. So those will get deuterated. And even the substrate. So if you talk about substrate water exchange, like substrate is what it acts on, it's like sub-second exchange times.
Oh,
so
okay.
yeah, whenever we kind of do the final preparation step with the DTO buffer, we know those are deuterated, and then that starts to release the deuterium,
Okay.
like D plus instead of H plus. Mm-hmm.
All right, thank you. Question, Chen Young, you go ahead.
Hi, Yulia. Very nice talk. Thank you. Yeah. I get question for the last one. You had the ESIF data for the photosynthesis tool. I mean, the bottom left is the x-axis is the pixel. It seems the C2 is the shift. Is the
Yes,
it's not
this is
the
just a
energy.
pixel. Yeah, so we are only starting like full data processing. Of course, it will be
Okay.
calibrated by reference. Yeah, those are just detector pixel number,
Okay.
not energy. But it can be calibrated because we have reference recorded. So they had a very good sample positioning system. So
Okay.
when we record the reference, we know we put it exactly in the same position as where we measure sample. So there is a system for that.
That's really
So
impressive.
basically, yeah, so we can, this is measured exactly at the same position relative to the spectrometer and beam as the PS2 data.
Oh, that's use the JS in the box, right? The Scott made the spectrometer. You bring the whole box there.
Yes, it's still in my suitcase because I still has not got back. Yeah, so this is the spectrometer, and it's been commissioned at APS, and this is a germanium 333 reflection. So germanium crystals that we bought, cut from ingot, we estimate this energy resolution. And again, like those dispersive spectrometers, they seem to have the highest energy resolution compared to other von Hammers where you have to focus multiple crystals into the same line. And this is the characteristics. Yeah.
Thank you. Excellent.
Yeah, it barely fits into the biggest suitcase I have.